Hello everyone, welcome to Assis Biology. In this video, we are going to discuss about apomixis, polyembryony and artificial hybridization, and this is the sixth part from this chapter, sexual reproduction in flowering plant. If you have not seen the first 5 parts, I will upload the link in the description box so that you can watch the video. Before starting this video, let me introduce myself. I am Ashish Kumar Das, a student of MSc Animal Biology, University of Hyderabad. And if you are new to this channel, please subscribe this channel and press the bell icon for getting more updates. Now let's get started. So first of all, what we will discuss today, we will see what are the special mechanism of reproduction. As you know, every reproduction or reproductive event is associated with gametogenesis, gamete transfer, etc. But we will see what are the unique features, that is the special mechanism apart from the main line. Okay, then we will see what is apomixis, that is also a special mechanism, and how it is occurring. We will see polyembryony, how it is occurring. We will see the examples of both apomixis and polyembryony. We will see what is apospory and apogamy. These are two important terms, apospory and apogamy. Then we will see what is artificial hybridization and how it is performed, and many more. First of all, apomixis. So apo means without. Okay, so this is without, and mixis means the mixing of two gametes. So without mixing, apomixis means without mixing. So this is a asexual mode of reproduction, but it mimics the sexual mode, that is it copies the sexual mode. If you see the result, it seems like it is a result of sexual reproduction, but in reality it is a asexual mode of reproduction, that is the mimicking the sexual mode of reproduction. Then what is the meaning? That is the seeds are formed without fertilization. This is called agamospermy. Okay, you have to remember the term agamospermy. The seeds are formed without the fertilization. In apomixis, the seeds are formed without the fertilization. As you know, mixis means fertilization, so the seeds are formed without the fertilization. So apomictic seed can be performed in two ways. You have to remember the way. What are the two ways? First of all, diploid egg, that is without meiosis, can be formed and it will develop into embryo. Okay, so as you know, the egg are always haploid. Being a gamete, it is haploid in nature, but here meiosis will not take place. So from the megaspore, from the megaspore mother cell, gametes will not be formed, or the spores will not be formed, and the egg cell will be diploid. Okay, that is with ploidy to end. And another mode is the cells of nucellus surrounding the embryo sac, that is the nucellus cells are the nutritive cell. They are diploid cell, but they will form the egg. Okay, and they will be pushed into the embryo sac. The example is citrus and mango. So what is apomixis? We discussed. What are the two ways in which apomixis can occur? We discussed, and the example we discussed. Okay, so you can see the diagram. In the sexual mode, you can see from the megaspore mother cell, as I have already told, the 4 megaspore are formed. From them only one will remain active, another 3 degenerate, and what happens, these are the degenerating, and cell specification is taking place, embryo sac is forming. Okay, and double fertilization, that is singamy and triple fusion, is taking place to form the embryo. However, in the aposporus or in apomixis, you can see what happens. The one of the initial aposporus initial, the red in color to distinguish, you can see it is remaining in the diploid state. Okay, and then it is forming the aposporus embryo sac, that is the diploid embryo sac. Okay, and from that what is happening, the fertilization is not necessary here, because we know the fertilization is to ensure ploidy, but here the ploidy is already 2n, that is the ploidy of the aposporus embryo sac. So here the seed is being formed from the embryo sac or from the embryo. Okay, and this is a asexual mode of reproduction, as you can see, because there is no gametogenesis. The gametogenesis is there, but no gametogenesis, that is diploid in nature, and no gamete transfer. Here it is developing as a sexual mode of reproduction, but it is mimicking the sexual reproduction. So a sexual mode mimicking the sexual mode. Okay, so apomixis, here asexual, you can distinguish from this diagram. Now come to polyembryony. So as the name suggests, occurrence of more than one embryo in the seed of a ovule is called polyembryony. And what are the regions for which it is occurring? That is from more than one egg in the embryo sac. Okay, more than one embryo sac in the ovule. As you know, in the ovule there are usually one embryo sac, that is the female gametophyte. But if there are more than one embryo sac, then there will be more than one embryo, and polyembryony will take place. Okay, this is also called as polyspermy, because embryo will leads to formation of seed also. Okay, so other cells like synergid. So the first two we have discussed. Other cell like synergid develop into other cell like synergids or cell from the nucellus may develop into embryo. Okay, and it is common in citrus fruit. Example are lemon and orange. Example is always important. The example are citrus fruit, that is orange and lemon. Other example are onion, mango, groundnut, etc. So polyembryony is most of the time it is associated with apomixis, that is both of them occur simultaneously in a plant, both are evident. It means if apomixis is taking place, there are more chances of polyembryony. So you can see here in the diagram, this is a polyspermy, polyspermy or polyembryony. Here you can see what is called polyspermy, because more than one seed will be formed here. Here you can see there are more than one embryo. The normal embryo is there, and there are nucellar embryo. These things are nucellar embryo. Okay, so here polyembryony will be there. Okay, so in this embryo you can see this is polyembryony in citrus fruits. Now you can see the diagram, polyembryony, embryo sac in Ulmus glabra. Okay, this is the name of the plant. Here they are zygotic embryo. We have seen the cell other than the egg cell may form into embryo. So here the antipodal cell, as a general rule, the antipodal cell must undergo, that is the degeneration, to provide nourishment to the developing embryo. But here the antipodal cell are developing into embryo. So this is also a type of polyembryony, and this is occurring in the Ulmus glabra. You can see the name Ulmus glabra. And this is the more than one embryo is formed due to antipodal cell, involvement of antipodal cell to form embryo. Now come to apogamy and apospory. Okay, so apogamy, apo means without, I have already told. So apogamy means without the gamete formation and syngamy. So the development of haploid sporophyte without gametes and syngamy. Okay, so how it is occurring? It is occurring in the means it is occurring without the gamete formation and syngamy. So example are Phonaria. Okay, Phonaria is a moss. Then both gametophyte and sporophyte are haploid. This is the important character. Both gametophyte and sporophyte are haploid in the apogamy. Then in apospory what happen? Without the meiosis, that is the sporic meiosis, that's what is called apospory, without the sporic meiosis. What happen? The diploid gametophyte, development of diploid gametophyte is there without meiosis. Okay, as a general rule, gametophyte should be haploid, but here the gametophyte are diploid because they are developing without meiosis. And spores from the vegetative and non-reproductive cells. So from where the gametophyte are developing? From the spore of the vegetative or non-reproductive cell of the sporophyte. As you know, in a sporophyte there are generative cell or sporogenous tissue, and there are vegetative cell and non-reproductive tissue. So sporogenous tissue will give rise to spores. But here is a anomaly or is a abnormal rule in apospory. What happens? The non-reproductive or vegetative cells of the sporophyte will give rise to the formation of diploid gametophyte. Okay, so here both gametophyte and sporophyte are diploid, and in apogamy both gametophyte and sporophyte are haploid. Okay, example is Anthoceros. So apogamy, apospory, both of them are both of them sometimes students are getting confused, but you have to remember the differences. Okay, here the gametophyte, sporophyte, ploidy level, how it is developing. Now artificial hybridization. So as you know, hybridization is formation of hybrid or making hybrid, that is the combination of desirable character from two different individuals. So when you are doing this artificially by human procedure, this is called artificial hybridization. So what happens, from the knowledge of pollen pistil interaction, scientists or plant breeders have worked in the field, and they manipulated the knowledge, and they tried to fuse even incompatible pollen to obtain the desired hybrid. So incompatible pollen means pollen from the different species to form the desired hybrid. So what are the advantages of hybrid? That is, two favorable characters or advantages from the two different plant can be combined to get a new plant, and this will lead to more yield and more nutrients in the fruit. Okay, or more growth of the food. Then major approach of crop improvement program. Artificial hybridization is a major improvement of crop improvement program, and tremendous research is taking place in this field. Okay, then what happens, how it is taking place? That is crossing two different species. This will be different DIFFPR into different species and different genus to obtain the artificial, that is the hybrid individual with desired character. So crossing two different species or from two different genus. Okay, then pollen from the selected plant is deposited on a stigma of selected individual. So if you have to choose a male parent, that is the source of pollen, and a female parent, that is the source of the stigma, and what happens, that is the source of ovule. Then what happens, we have to deposit the pollen grain, and it will lead to the formation of the desired plant. Okay, so stigma is prevented by contamination. So what you have to do, you have to cover the stigma. After you are providing the selected pollen grain, you have to cover the stigma such that the unwanted pollen grain will not be laid on the stigma. Okay, then these are the processes associated with the artificial hybridization. So emasculation, anther are recovered removed from the bisexual flower before maturation. Okay, so here you can see the diagram. The anthers are being cut with the scissors. Okay, and pollen is dusted from here. These are used to dust the pollen grain. Okay, so emasculation means the removal of anther from the bisexual flower before their maturation. Then bagging, that is emasculated flower must be covered with suitable bags to prevent contamination from other pollen grain. And tagging, the leveling of the plant, means when you are doing for the number of plants, doing the process for a number of plant, artificial hybridization for a number of plants, what will happen, you have to cover with suitable bags. Again you have to provide the tag, that is with the name of the plant, date on which you have emasculated, you have bagged, you have tag, etc. So tagging is the providing suitable name or labeling the name with a sheet of paper, or with a paper sheet or a sheet of small paper. Okay, then when stigma become receptive, what happens, the mature pollen grain are dusted after emasculation, bagging, tagging. Mature pollen grain are dusted on the selected plant and this on the stigma, and this will lead to flower, means this will lead to fruit development. Flower are bagged again and allowed to develop to fruit. And if the female parent produce unisexual female flower, what happens? The female parent produce unisexual or female flower, the flower buds are bagged before they open. What happens? Because if you are not cover them with a bag, there are more chances of contamination, so you have to cover them if the female parent is the, so both in both the cases, that is the unisexual bisexual flower, you have to cover them, but here after the removal of anther. But in the female parent you have to cover the unisexual flower before they open. Okay, this is the important difference. Thank you.